Good afternoon, ladies and gentlemen. I would like to start by acknowledging the summit's organization for the invitation and opportunity to participate in the Global Summit on Animal Science and Veterinary Medicine, Tokyo, Japan. Today, I will be presenting part of a broader research project entitled Microbiological Monitoring Program in an Artisanal Dairy, a 2-year exploratory study case. The production of traditional cheese is an important part of the Portuguese cultural heritage. The richness and specificity of such products reflect their artisanal manufacture practice. In a cheese workshop, while autochthonous microbiota is responsible for fermentation processes required for cheese production, several sources of contamination can be identified. So cheese can be a potentially hazardous food as it is a ready-to-eat food, not requiring a heat treatment before consumption. Because of that, microbiological monitoring of the food processing environment is of utmost importance in preventing final products contamination and potential consumers' foodborne infection. Cheese can get contaminated with foodborne pathogens that are able to resist for months or even years in the food processing environment in biofilms. Final products get contaminated through cross-contamination, and cyclotrophic pathogens such as Listeria monocytogenes might be worrisome, as they might endure long refrigerated shelf lives as the ones presented by several cured cheeses. Regarding these facts, the main aim of our study was to assess the microbiological monitoring program of a dairy industry located in Setúbal's district, Portugal, from 2020 to 2021. As for sample collection, food samples collection was performed according to international standards, and 180 samples were collected. Food samples included raw milk, butter, fresh and cured cheese. Environmental samples collection was performed according to international standards as well, and included swabs of food contact surfaces after cleaning and sanitizing procedures were applied, and it also included food handlers' hand swabs after hand washing procedures. International Organization for Standardization methods were used for food contact surfaces, and hand swabs microbiological analysis as mentioned in this slide. And results interpretation criteria considered both the European Regulation 1441 of 2007 and following amendments on microbiological criteria for foods, as well as the microbiological guidelines for ready-to-eat foods and processing environment published by the Portuguese Health Institute in 2019. A database was constructed combining environmental and food samples results of microbiological testing. Microbiological evaluation for process hygiene and safety indicators revealed that approximately 37% of the tested samples were non-conforming, namely 17% of the food samples and 20% of the environmental samples. Of the 30 non-conforming food samples, 17 presented countings above the criteria established for Escherichia coli, 7 were above the limit for coagulase positive staphylococci, and 1 was positive for Listeria monocytogenes detection. It is noteworthy to mention that Salmonella spp was never recovered. Also, 5 fruit samples presented countings of Enterobacteriaceae above the limit. In environmental samples, Enterobacteriaceae quantification reached 5.2 log CFU per 10 square centimeters. It is important to highlight that most of the non-conforming environmental samples belong to the hand swabs, although Enterobacteriaceae countings presented higher values in food contact surfaces. Additionally, Listeria monocytogenes was not detected in environmental samples as well. These results could be related with poor general hygiene procedures and practices with fails such as improper cleaning and sanitizing of the food-related environment and incomplete hand washing procedures. Considering the hand washing procedure, an incomplete procedure was noticed with improper use of soap and of hand drying methods. Considering premises hygienic procedures and practices, fails such as improper cleaning and sanitizing method application and insufficient contact time of sanitizer with food contact surfaces seem to explain the results. Also, the occurrence of pathogens could be due to the use of closed equipment systems and to the lack of rotation schemes considering sanitizers used in the food processing environment. Taken together, our results reinforce the need to refine and improve the hygiene procedures and practices in the Assess Daily workshop, as well as the importance of establishing and implementing appropriate and thorough corrective actions after non-conforming microbiological monitoring results. The authors gratefully acknowledge CISA for funding this project, as well as my alma mater, FMV, at Lisbon's University. I am now ready to address any questions. Thank you very much.
Thank you for that insightful talk, Doctor. As the speaker is not available right now to answer your questions, now moving